using an approach with light. So I would like to give you some ideas of what do I mean by this. In the presentation, I'm going to present the hypothesis that transcranial infrared photons in a dose-dependent manner can upregulate cytochrome oxidase. That's the mitochondrial enzyme that is our molecular target. And by doing that can result in increased cerebral oxygenation and neurocognitive enhancement. I will tell you a little bit about the molecular mechanisms. All of these studies in terms of molecular mechanisms we work out in animals. However, today I'm going to present you our non-invasive human physiological studies demonstrating these mechanisms for the first time. And I will show you about cerebral cytochrome oxidase measurements done in collaboration with bioengineering and hemodynamics, and then some of the cognitive effects in humans. One of the important things is, as we all know here in this group, is that oxygen metabolism is critical for brain function. So based on Professor Rattan's talk today, the approach that I'm following is trying to be friendly to oxygen metabolism. And one of the things that we have done is develop infrared laser as an approach to provide photons that can be absorbed by cytochrome oxidase, and in this way accelerate oxygen consumption. This mechanism is favorable because the oxygen is reduced, so it actually minimizes free radical formation, even though there is an initial increase, it's very low, and it increases metabolic energy. And actually the levels of the light that we use are very small in terms of the milliwatts per centimeter square. And however, because we allow sufficient time in terms of minutes of exposure, we can obtain high enough energy levels that can reach the brain. And this was an important issue when we did this in animals. We could measure directly how it went through. In humans, we had to do some extrapolations, but we were able to measure this now directly. So I'm going to give you a little bit of backgrounds from our studies. I would say that our key study was in 2008. That was the first time that I came to these meetings. But at that time, I was talking about a pharmacological approach to increase mitochondrial respiration. So now we found a way to do the same thing, but without having to use drugs. And it's a very non-invasive, safe approach. In animals, we've shown that it increases cytochrome oxidase by directly measuring this with quantitative histochemistry. And it also stimulates superoxide dismutase, so it acts as double role. It increases energy metabolism, while at the same time, it's a neuroprotective intervention. And in the 2012, we developed this in animal models targeting the prefrontal cortex, and we, for the first time, then show that you can affect memory function. That not only we're increasing cytochrome oxidase level, but by doing this, we were able to improve a prefrontal based cognitive response. And then by 2013, we were able to do this, translate this into humans by using an FDA-cleared device that I will introduce here. And we have follow-up from attention and memory studies to other types of cognitive functions, for example, what I refer to as executive functions. But also we've done some populations that some of them have depression, in which having a treatment that improves cognitive performance also improves some of their symptoms. And more recently, we extended to category learnings, and showing that depending on the kind of category learning, if we target only the prefrontal cortex, we stimulate only that particular type of learning, but not some other learning in the brain. If you don't remember anything from this talk, I would like you to remember these three words and the relationship between them. The important thing, because cytochrome oxidase is such a crucial enzyme in mitochondrial respiration. But the interesting thing is that it can absorb these near-infrared photons. So we're taking advantage of this biochemical property. So a little bit of background. Not all light is effective in doing this. What we found that is more effective is around 1,000 nanometers. So it's actually 1,064, the laser that we use. And we have tried different ranges. It starts working out close to the red. In other words, the enzyme can absorb those photons. So from the 600s to the 1,000. However, below that, it's not really effective because there are other so-called chromophores, like melanin, that absorb the light, so it cannot really penetrate the tissues. But also after 1,100 nanometers, we have water that absorbs this and produces heat. And actually, none of these effects that I'm going to talk about can be explained by heat. We've done the same experiments producing heat to the forehead and showing absolutely no effects on the same and measurements. There are properties that we take advantage of. The longer the wavelength, the more penetration there is in tissues. The longer the pulse, also the more penetration, and it penetrates more in the gray matter than the white matter. So our treatment is really limited to the gray matter of the cerebral cortex. And from there, it activates all the networks. And this is the particular device that we use. This is familiar to you. This is a caricature of this respiratory chain where you have some of this respiratory complex, and the molecular target is cytochrome oxidase. And in animal studies, we're seeing how these enzymes absorb the photon. You can see how the catalytic rate of the enzyme increases. You can do it in mitochondrial preparations. You can do it in brain homogenates. You can do it in vivo in rats and mice. And you will see here that it happens also in humans. But the interesting thing is that after you come back, for example, 24 hours later, there is an upregulation of the enzyme. So there is now more metabolically active cytochrome oxidase available after you do this. And we recently has worked out this simple model. And in the human, we've been able to demonstrate this, the increase in oxidized cytochrome oxidase, and also the increase in oxygenated hemoglobin. And basically, when the photons go here, the enzyme releases some of the nitric oxide that is in the catalytic pocket, and that produces a local small vasodilation. The enzyme then starts, in the oxidated form, starts reducing oxygen to water, and the process of proton pumping is activated, and has a consequence of these protons moving through the ATP, the ATPase, the process of ATP consumption. And simultaneously with these, we get a slow hemodynamic response that follows the increasing oxygen consumption. One of the things that we found early on is that these phenomena, for example, if we measure oxygen consumption or if we measure cytochrome oxidase activity, there was a hormetic response to this light. And it was around 1 joule per centimeter squared. That's a measure of energy, power by time, where we found the best effects. So this hormetic peak is what we had tried to target in all of our studies now. So for example, in rats, we have to, because only about 5.8% goes through, we have to do like 21 times more of the irradiance in order to get the right peak. In humans, it depends on where you look at, but at the frontal bones, only about 2% of this light actually goes through. So we have to do, if we want to target, for example, 1, we need to do 60 times more. And you can see here that 1.2, by, by using 60 joules, we can reach that amount of energy getting into the brain. This is the instrument that we're using for the human study. Like I mentioned, it's FDA clear. It's actually, there is approval for other indications by FDA in much higher watts. What we normally use is about 3.4 watts in this instrument, but when it's using 18 to 20 watts, it's used for pain relief because of that hormetic dose response that I show you. At the lower levels, it stimulates brain or neural function, but at the very high levels, what it does is inhibits the nerve terminals. So it's used for pain relief as an analgesic approach. This is a study using in vivo a technique that is called functional near-infrared spectroscopy. One of the advantages of this technique is that we can dissociate the oxidated hemoglobin and the deoxidated hemoglobin, as opposed to, for example, functional MRI, where you get these bulk signals that are a combination of these. And in this study, in collaboration with the bioengineering group, we stimulate for 10 minutes, and we actually only measure at the end. We do 55 seconds stimulation and 5 seconds of recording. And this is a technique, just like spectrophotometry, that is used in biochemistry, but it can be used non-invasively through tissues. The advantage of this is that as soon as we start stimulating, if you compare to baseline, you can see an increase in oxygenated hemoglobin. If you count here 1, 2, 3, 4, and notice that 4 minutes is what corresponds to the predicted peak effect in the brain. So this was a physiological confirmation that our in vitro work is actually guiding us in the right direction. This is in the right and the left hemispheres measured with different channels. And a similar thing happens, but as you can see, if we continue with the time of stimulation, the effects tend to go down. In terms of light, the time that this happens is really a dose response, because the power here is constant. We have a constant power of 250 milliwatts per centimeter square, and we only change the exposure time. So what you're seeing here is a dose response. Interestingly, when you stop the stimulation, there is a continuation of this response. In these studies, with this number of subjects, we've done 2 groups of 9 subjects each. We only measure for about 6 minutes, but in some of the subjects, we measure 20, 22 minutes, and it's continue to have increased levels of oxygenated hemoglobin happening. Now we develop with this group another way to look into this. These were different subjects, and this is a somewhat different technique that is called broadband near-infrared spectroscopy. The advantage of this technique is that it allows us to measure the concentrations of cytochrome oxidase non-invasively through these forehead recordings. If you notice down here, these are cytochrome oxidase concentrations, and what we are actually measuring, the enzyme can be present in 3 redox forms. The oxidized form, which is the one that has affinity for oxygen, and there is a mixed state one, and the reduced enzyme that is they have the least affinity for oxygen. So what we see is that as soon as we start with the light, there is an increase in cytochrome oxidase. And these are these red lines that you see here. The blue lines that are shown here on the bottom refer to a so-called sham or placebo situation. In other words, these subjects, in some experiments, they get the real laser light, but in others, they get a sham light that doesn't produce these effects. And but about 2 minutes, you get significant differences between placebo and the laser stimulation with the infrared. But let me tell you about some of the other effects. You can also measure the hemoglobin oxygenated and deoxygenated concentrations, and by about 3 minutes, then you see changes in oxygenated hemoglobin. So what this implies, and we tested this in some other ways, is that the initial event after photon absorption by cytochrome oxidase is an increase in oxygen consumption, an increase in the oxidated enzyme level that then triggers a hemodynamic response that follows. And but about 4 minutes, then the levels of deoxygenated hemoglobin also start going down compared to placebo. So at the same time, so what this is doing is increasing cerebral oxygenation through a direct action on cytochrome oxidase as a photon acceptor. As far as we know, cytochrome oxidase is the major, if not the only, intracellular photon acceptor for this particular wavelength. And now I'm going to show you some of the results that are not near-infrared spectroscopy. These are studies in which we look at the EEG. So the subjects are receiving the same kind of stimulation. And what we're doing here is you simply measure what I refer to as the EEG rhythms, the frequency of oscillations. And one of the things that happens, and this is so-called a power spectrum, when you're presenting the stimulation, you have a very dramatic increase in alpha waves. This is a particular recording from the prefrontal areas, in other words, by looking at electrodes in the forehead. To very lesser degree, we have beta and gamma activity. This is actually starts out in the prefrontal cortex, but as a function of time, you can see the same changes happening in networks throughout the brain, from frontal, then to parietal cortex, then to temporal, and it starts on one side. We're doing a stimulation on the right side, and then it becomes bilateral. So even though you're targeting one particular region in the brain, this executive region, you can get the whole brain engaged. I'll let you read this for a moment. These are the parameters that we have found to be optimal in our studies. If we were testing different situations, there may be other parameters that may be, so one will have to adjust the protocol depending on the outcome measured. But like I told you, in this particular device, we only use a very small level of power for at least 4 minutes per side. So we usually stimulate 2 sides in the forehead, and we normally alternate between the 2 sides. So it's what is called fractionated, like 1 minute in one side, then 1 minute in the other side. It's known from our animal studies that this fractionation improves the response of the tissue. And it's a fairly large stimulation area. This is a very critical issue. Even though we're using lasers, we normally, like this one, can have very small diameter. This is, from a practical point of view, is not a good idea in terms of the brain. In other words, we need to stimulate a large enough volume of tissue in order to get these effects, especially if we're going to get cognitive effects in humans. So it is important. These kinds of lasers are not applicable. You require one that will have a much wider area of delivery of the energy. And again, these are the calculations that we use originally were based on measuring cadaver frontal skulls, these 2%, and now we know that these measurements appear to be effective. Again, what I'm going to show you here, I mentioned the previous studies. All of those previous studies in humans, I did with healthy young college students over there in Texas. So what we decided to do later on was to use, doing a small study to look at older people. So, and we use this, the results from this study to get NIH funding, and now we have an NIH grant from National Institute of Aging for doing a larger scale study with 200 subjects. But I will present you basically the pilot study that we did with these 12 people, trying to replicate the same kind of things that we saw with the healthy young adults. But one of the differences in this study is that the previous studies, we have done only one session of the stimulation, which lasted 8 minutes. In this case, we did one session per week for a total of 5 weeks. And we use this same task that we used before. This is a sustained attention task, the psychomotor vigilant task. This is a task that is used by astronauts and the military to monitor sustained attention. We use a delay match to sample memory task. It's a type of working memory task. One thing in common between these two tasks is the high reliance on the prefrontal cortex, in particular the right hemispheric prefrontal cortex. So that's what we use as a target. And then we look at the acute and the chronic effects during this period. This is to show you some of the parameters that we measure. The reaction time. Reaction time is a fairly static parameter from a cognitive point of view. It's not easy to change your reaction time. So if your reaction times can go down, that's the beneficial effect. You can make faster responding. And you can see that after acute or chronic administration of this, we get significant decreases in reaction time during this sustained attention task. If you also, for example, look at the number of lapses, it is possible in this task, especially this older population that we had, that they will fail to make the response during the time constraints of the task. These are also, number of lapses are significantly improved by having the laser stimulation. And the number of correct responses is increased. But notice that there's not much change, at least on averages, between acute and chronic. So one of the things that we done is to try to understand this phenomenon a little bit more by looking at in more detail at those response curves. So here is similar data, but using normalized, the percent change, normalized with respect to each subject. And as you can see, from baseline, these reaction times go down, but by about 4 or 5 weeks, there appears to be a return going back towards the baseline. It doesn't go back yet. It's still effective, but at this point, we are not sure whether there is a cumulative effect and at what point is the optimal point to stop this. Similar kind of effect is happening here, but there's more variability with the correct responses. So one of the things that I'm going to show you are raw data now in the next slides, which what we have tried to do here is just simply look at the data. And for example, this is now the raw score. Over here is just a simple linear regression. But the actual average, if you look at the green line here, the actual average also shows this bump in the last week. And if you look at individual subjects, the majority of them show this particular increase in the last week. By the way, we had 2 subjects with 90 years of age. And in both of those subjects, in each one of these measures, they improved by about 30%. All of the subjects, the 12 subjects, they all had subjective memory complaint. And one of the 90 years too was clinically diagnosed as having mild cognitive impairment, and there were 3 others that had mild traumatic brain injury, and they all benefit from the treatment. Similar thing happened here. This is a correct scores, but the raw score against, and there was a tendency that these scores were going down for most of the subjects in the last visit. One of the things that we did a little follow-up, we tested a subset of subjects using functional near-infrared spectroscopy, where you have multiple channels in the area of the prefrontal cortex, and you can have them do the same two tasks, the psychomotor vigilance and this delay match to sample task. And after, and compare before and after the laser stimulation. And what you can see is that there is now a greater engagement of cerebral oxygenation that happens when they do the task again after they get the treatment. And notice also that both hemispheres are activated. So even though we stimulate on the right side, through the commissural connections, we get response on the other side of the prefrontal cortex. So my conclusions here is that transcranial infrared laser stimulation could be used as a non-invasive and efficacious approach to increase cerebral cytochrome oxidase and oxygen metabolism and support neurocognitive functions in humans, including people with elderly people with cognitive decline. And we find this safe. We've done over 300 subjects so far with the laser treatment. And we have not a single, not even one of those that happened today, these adverse events reported. So it appears to be a very safe situation. And most of these subjects were healthy college students, so we may be able to find more adverse reports when we start looking at the older population. Your imagination is the limit to what you can do with this. Right now we're focusing on the forehead. The reason we're focusing on the forehead is that we don't have hair. And the pigmented hair absorbs some of this light energy. So by just working on the forehead, we target this prefrontal area. This prefrontal area is the executive in the brain. It can direct and orchestrate responses throughout the brain. And so far, we also measure people with different pigmentation. The amount of pigmentation in the skin doesn't seem to make any difference in terms of the dosing. Even though with this spectroscopy, we can actually do a dose response before we do a treatment and individualize the actual level of the treatment to that particular individual that we can follow up then during the chronic treatment. Just want to thank you, my faculty collaborators here, Dr. Liu and Tian from Texas Arlington. These are the bioengineers who work on tissue-light interactions. Barrett and Blanco, former trainees of mine, and a number of co-authors are primarily students in working in this for the past 10 years. And these are some of the key references. Thank you very much.
Not
Well, let me answer the two questions. The answer to the first part of the first question actually answers the number two. This is similar to electrical and magnetic stimulation in the sense that the word transcranial is used, and in the sense that we're talking about brain stimulation. However, it's completely different from a mechanistic point of view. When you do electrical stimulation or magnetic stimulation, you spend energy. When you're stimulating the tissue is using energy. When we do this, we are providing energy to the tissue. We are providing energy through cytochrome oxidase. Cytochrome oxidase is increasing not only oxidation, but increasing ATP levels, is moving on the oxygenated hemoglobin. Normally, when you do, for example, electrical stimulation, you get the neurons to show more activity. They expend more energy, and as a consequence of that expenditure, then you have a hemodynamic response. That's not what's happening here. We are increasing energy levels due to the stimulation. So because of this is fundamentally, mechanistically different than transcranial, magnetic, or electrical stimulation.
That was my question.
Well,
my question had to do with the area of
well, that's the second part of the first question. So the second part of the first question, what is the area stimulated? The area stimulated is similar to what I showed you there, in that I have the area of what is about 4.2 centimeter diameter. So if you look during the stimulation, that area underneath the laser light, like you see, is collimated. So only those neurons are actually receiving it. However, as soon as you engage the brain in any other task, this starts moving. And you get, for example, in those EEG effects, we done the E-Loretta analysis, and you can see effects then throughout the brain. So the initial stimulus is under the laser, but we now we have it is possible to develop models similar to the ones that you use for intensity. But the problem with electricity, when you put electricity, it's going to move throughout the entire body. This doesn't happen with light. Light is absorbed. It just doesn't keep on going somewhere else, because the electricity, you're providing electrons. Here we're providing photons that are absorbed by the tissue. So it's a self-limiting process. So the second part of the question then is related to that phenomenon. If you have a context, he's asking what a context. You can do this stimulation in two ways. You can just simply stimulate before you challenge the brain, but essentially you are charging up these regions, providing energy reserves, and then when you challenge them, then these regions can more effectively cope with the challenge. Alternatively, you can have, like what we did with the depression study, we can have people do a task that we know diminishes depression symptoms, which is attention bias modification, people modifying their bias from looking at this and saying it's glass not full, half full, but half empty. Everybody who's depressed is going to say half empty. So one can change this by focusing on neutral as opposed to negative things. Then when you do it at that point, you facilitate that process. So whatever ongoing challenge you have, if you provide more energy resources, you are able then to benefit more. However, this can be done offline as opposed to the transcranial electrical or magnetic stimulation that is more effective than if it's done online. In other words, because you're just increasing a threshold. No, you can do this ahead of time, and you also can do it after the task. For example, in our memory tasks, you can do this learning. You can stimulate after that, because after learning, there is a period of memory consolidation. And you can facilitate, for example, we've done this with a fear extinction. That is a form of memory for reducing anxiety disorders. So it doesn't have to be online. It can be context independent. And you can bring then the context later on.
Other question?
Cerebral blood flow go up?
Yes, in this technique, what is called the difference between oxygenated hemoglobin and deoxygenated hemoglobin is an index of cerebral blood flow. So it goes up, but it goes up as a consequence of the increase in cytochrome oxidase concentrations, not as a direct result of the stimulation.
And if I put this laser device on my chest wall, can I reach the heart?
Yes.
Is anyone doing that? Is anyone looking at
There is in animals, yes. There is a group in Israel that is doing this in animal models. And not only directly into the heart with heart attacks, but even in a more science fiction way, he's stimulating long bones that have stem cells, hematopoietic stem cells in the bone marrow, after producing in a rat model or a porcine model a heart attack, and like in the hips or the long bones, and he can tag those cells and see that then they go. He's just facilitating the response of these stem cells. They go as a repair mechanism, then end up in the heart. This has been done so far by this group. So the applications are not just limited to the brain, but it could be directly in the heart. Because as compared, for example, you couldn't do that with electrical or magnetic stimulation. Will be completely disruptive to the function of the heart. Here, you're just providing energy, chemical energy. You have a question?
Have you contemplated to study intercellular communication from the cells that have cytochrome oxidase activated, maybe collagen junctions, which are very prominent in the brain, and that's when maybe it populates to nearby areas?
Yes. I would like to cooperate with somebody that has the tools to do that.
Animal models.
Yeah, right now I've been trying to translate this into human applications. That's been my primary focus.
We're running a little late during the break. We're running a little late. I want to make sure we give time based upon.
Thank you.